Alrighty, let's fight off some infections. So, our immune response is when foreign cells are detected, so foreign also including viruses or sometimes some small fragments that will elicit a immune response. You also can elicit a immune response or activate a immune response from chemicals that have been absorbed into the body. So this is why we tell you to do all that safety stuff in lab, so we don't end up irritating your body. Okay, so with immunity, we are guarding against anything that is foreign, and our response is with our immune system. So back to Cornyville. So our immune response is based off different immune cells and different proteins. So this is where I start referencing different things as ways to kind of remember what they do. So welcome to lymphatic system high. The rival is pathogen high, and those are agents capable of producing disease. So that's what pathogens are. So it could be virus, it could be a bacteria, it could be fungi. Anything that is a foreign agent in our body will elicit or start a immune response. Sorry if you get any flashbacks from high school. Oops, oh well. Okay, so we have 3 lines of defense in our lymphatic system. We have the first line of defense, that's the skin and mucous membrane. So outside surfaces of the body or exposed to outside surfaces of the body. And this is a physical barrier to keep the pathogens out. Second line of defense, this is innate immunity. So this is where the immune cells recognize that this pathogen has entered the body, but it's non-specific. So basically their job is to kill anything that is foreign to the body. So it's just a generic, you all can't sit with us. So if it doesn't belong there, that second line or innate immunity, so the immunity you're just naturally born with, will attack anything that's foreign. So even though it might be someone else's kidney that you got as a organ transfer, same thing. That glycocalyx is different. So this is why people who get organ transplants, they're also given immune suppressors so that their immune system doesn't attack their new kidney. The third one is adaptive immunity. So this is the immunity that is developed specifically for that specific pathogen. So that's the reason why we get vaccinated, is because it is developing a defense against a specific something. So against measles, against COVID, against the flu, all those vaccines, that's what they're doing. So you can have a built up defense against that specific thing so that if you get it, you are already prepped and ready to go to fight it off. So you end up either asymptomatic completely, or you have a better shot at surviving, and you might have some symptoms, but you have a just better shot at surviving just because you are easily getting over it, just because you already had this defense system set up. So with our adaptive immunity, it is you specifically can't sit with us. So it's focused just on certain pathogens. And again, it's because they've been activated against that pathogen. So with our lymphatic system actors, or our what their jobs doing in our school, so I broke them up. So we have our upperclassmen. These are our lymphatic cells. So those lymphocytes, which is one category of our leukocytes, so our white blood cells, those differentiate or become even more specialized. So they have a kind of extra step of maturity. They are kind of the more powerful of your white blood cells, doing the job of killing off anything foreign. So that's why they're in their own special class. Next one will be the freshmen, so just our regular leukocytes. So what does the rest of our white blood cells do? And then this one, I call it journalism, and you'll see that why later, because their job is to communicate to the immune system. And so these are antimicrobial proteins. So some of these could be, for example, be your antibodies, or other proteins that their job is to target any kind of foreign pathogen. And when they target it, they're able to spread the message to the rest of the immune system, as in, hey, this person shouldn't be there, or this pathogen shouldn't be here, attack it. Okay, let's get started. So with our upperclassmen, these are in lymphatic cells. So we have our antigen presenting cell. This is a category of cells. So we have different cells that are in there. Their job is to, and they also fit in the phagocyte section. Their job is to literally engulf the foreign pathogen, digest and break most of it up. So that's also going to neutralize it. But then take a little piece of that and display that foreign antigen onto their surface to alert the immune system as in, hey, we have a problem. These next three, natural killer, T lymphocytes and B lymphocytes, these are our specialized lymphocytes and they have their own specialized job. Phagocytes, their generic thing is just to eat up stuff, and their job is just to eat up anything foreign, and they're just not in this also other category of antigen presenting. So they could only job is to eat stuff and they're done, versus antigen presenting will eat something up but then have that last little bit, that foreign antigen, that foreign glycocalyx, to kind of wave the flag as in, hey, immune system, come do something. Okay, and the last one are structural cells. So earlier we talked about our different lymphatic tissues and lymphatic organs. So we do have some structural cells that will help with those organs as they function to provide structure. Okay, so focusing on our lymphatic cells, so we have our powerful lymphocytes, so our NK, T, and B cell, our phagocytes, which are macrophages, as well as our dendritic cells, so you remember that from the skin chapter. Structural cells are also called reticular cells. So they're in specific places such as your lymphatic organs, your skin, as well as mucous membrane. So specific places where they are highly likely to interact with a foreign pathogen. So let's start with the antigen presenting cell that I named Gossip Girl. So their job, again, they're going to go through phagocytosis. So the category that this is, the cells are in this category. We have our B cells. They're going to do this. So they help to activate your T cells, by the way. Your macrophages, your reticular cells, and your dendritic cells, all of them will gulp up the foreign pathogen, take that foreign glycocalyx, display it on the outside of their cell, and therefore able to activate other cells. So, for example, your T cells will be activated during this process, and therefore they can start their immune response. So basically their job is to collect dirt. Hey, we have this foreign pathogen. This is who specifically this is. All y'all, I need y'all to mount up and go get this person. Basically spreading a bunch of gossip. Okay, so the reason why we have antigen presenting cell is because some of our immune cells cannot recognize a foreign antigen by themselves. So they need those antigen presenting cells in order to do that. So there's our list. Okay, so in order for these antigen presenting cells to do their job, they have what's called a major histocompatibility complex. So this is kind of like a flagpole for displaying the type of cell. So your glycocalyx will be as part of this, this is a part of your glycocalyx to identify, hey, this cell belongs to us. But for your antigen presenting cell, they'll stick this extra foreign piece off of that flagpole and showing that, hey, there's a foreigner among us, so we need to go attack it. So how this works, so our antigen presenting cell will encounter a foreign antigen, so it actually is able to detect the foreign glycocalyx. It internalizes it, so it uses endocytosis, so brings in a vesicle into the cell, digest it. So remember, we're talking about those granulocytes for our lymphocytes, so the ones that are white blood cells that have the little speckled appearance, yeah, they have enzymes to break down any kind of foreign pathogen. Okay, so it's going to take a fragment of the foreign cell and display it on that major histocompatibility protein. So that displayed fragment is going to alert the rest of the immune system. So for example, that T cell, it can read that major histocompatibility protein, and if it sees just the normal cell glycocalyx flag, then it's like, oh, okay, whatever. But when it sees that foreign one and it sees it twice, then it's activated as in, okay, let me go get this person. Well, pathogen. So here is a picture version for those who like it. So here is like for example, a pathogen, bacteria, whatever. So we have our endocytosis, our digestion with our enzyme. So lysozyme, just a special word for enzyme. So it's broken up, but part of that is displayed on that major histocompatibility protein, and then all this other debris is just released from that cell. So this little flag, that's going to stay on that protein to activate your immune system response. So as these antigen-presenting cells are just kind of circulating around, so they go through the different tissues and just move around, T cells can, again, they can read that major histocompatibility. If they see a self-antigen, so your own glycocalyx, whatever, sees a foreign one, it starts its immune attack, but it has to be activated twice. So your antigen-presenting cell, they're ones that are going to alert the immune system and mobilize them against that antigen. They can also secrete out chemical messengers called interleukins. And what they're going to do is they're going to help to draw in other cells to a foreign pathogen, as well as activate the immune system. So again, their job is to ring the alarm, if you will. So what cells start with this process? So first type of lymphatic cells, so our lymphocytes, again, we have 3 of them, NK, T, and B. The NK, these are called natural killer cells. So I call them the queen bee because of how it works. Their job is to attack and destroy the bacteria. So just kind of attacking anything foreign, because sometimes, you know, how some people can be in high school and their job is just to antagonize people. So they're part of what's called the innate immunity squad. So innate immunity is your natural born immunity. So they just target anything that's not one of you. So anything that's not foreign in general, attack and destroy it. So it could be bacteria, it could be a transplant tissue, it could also be host cells that have been infected by a bacteria, because once that cell gets, not infected by a bacteria, infected by a virus, so once that cell becomes infected, it will alert the rest of the cells around it as well as your immune system, so your natural killer cells will also go against it as well. So how does it work? It releases what's called preferrin proteins. So that's this little purple ring that's been formed here. So it creates a hole in your foreign pathogen. Then, so that's one set of minions. Then it releases granzymes. So that's the second set of minions. They go in that hole that our preferrin proteins have produced. And they essentially degrade and destroy stuff in that foreign pathogen until that foreign pathogen goes through apoptosis or kills itself. Yeah, I know. It's rough, but it works perfectly with that kind of queen bee type of chick that antagonizing everyone. And yes, that's why some kids, they eventually kill themselves. But attack until that foreign pathogen kills itself. Okay, sorry. It sucks, but oh well. Okay, so let's look at our T and B lymphocytes, also called the tag team champions. So, you know, that bromance that probably every school has. And just randomly, the picture, like, who are these dudes? That's actually Dwayne Johnson's dad, and then that's his partner that he would do wrestling tag team with. Okay, right now. Okay, so with our B cells, they are the antigen-presenting cells, so they're the ones that can actually detect a foreign antigen, and they will activate the T cells. Then, when the T cells are activated and they go through their process to start an immune response, one type of cell that they develop into is a T helper cell that can then stimulate the B cells to do their job. So their B cells are also going to make clones in order to fight off that specific infection. So both of these cells are in what's called the adaptive immunity, where they're adapting or specifically developing a defense against a specific pathogen. Okay, let's start with our T cells, which I call student council. These are your most abundant lymphocytes. Again, adaptive immunity, so going against a specific type of infection. And this is the cellular immunity addition. So in adaptive immunity, you have two different types. You have cellular, which is what the T cells would do, and you have your humoral immunity, which is what the B cells was going to do. So with our T cells, when it recognizes the foreign antigen that's been displayed on an antigen presenting cell, it has to be co-stimulated, so it has to be stimulated twice. And its job is going to create an army of clones to attack the current situation, but also create memory cells to remember that foreign antigen. So if you get infected with that same thing again, you already have clones ready to go in order to fight it off faster. So same thing with the vaccines. It will create a bunch of T cells specifically against that pathogen that you're being vaccinated for. Okay, so with our T cells, they are born in the bone marrow, just like your other white blood cells, but then they are shifted off to the thymus, so they get sent to boarding school. So that's why the T part of T cell. And then once they become immunocompetent, so able to recognize a foreign antigen that's been presented to them, and they don't react to a self-antigen, then they are finally deployed into your lymphatic tissues. So when they are in boarding school, they go through they possibly go through two different tests. So making sure that they only respond to a foreign antigen. So first thing is positive selection. So that is where they must develop the ability to detect the foreign pathogens off of a antigen presenting cell. If they do not react to a self antigen, then so self glycocalyx flag, whatever, if they don't react, then they're sent into the naive lymphocyte pool. So they haven't been exposed to a foreign antigen yet. So of course they wouldn't be possibly activated yet. But if they react to a self-antigen, so they have the ability to read a major histocompatibility complex, but they reacted when it was a self-antigen, so just a regular cell, then they go through negative selection. So this is when those T cells fail. So basically they get expelled out of school. So they either go through clonal deletion. So remembering our T cells, the way they're created is through just slight genetic differences, so you can detect different foreign antigens. And those T cells that fail, they're phagocytized. So that cloned section, they're just basically eaten up and destroyed. They're also either made inactive, so they're not given a certain cell signal that would allow them to stay alive. So let's look at the ones that actually pass the test. So those T cells are deployed into different lymphatic tissues and organs where they can be ready to be activated. So let's say they do actually get activated. In order to do so, you need an antigen presenting cell that is displaying that foreign antigen on that major histocompatibility complex. So again, you have to co-stimulate a T cell, so it has to be done twice. When it does so, it will create a bunch of clones that will either become a T helper cell, a TC cytotoxic cell, or a TMT memory cell. So these are the ones that are just going to hang out in your bloodstream. So just in case you get infected again, then you already have a defense against that specific pathogen. So this is how this works. So after you have clonal selection, so again, this specific clone T cell that is reacting to that specific foreign pathogen, go through mitosis to make a bunch of the same clones. And these are all programmed against the same foreign antigen. Some are going to actually attack your current situation while the other ones become memory cell. Okay, so this is just, again, if you need pictures. So again, going into different types of cells after it's been co-stimulated. Okay, so let's look at what these cells do. So the first one, our T helper cells. So this is where our tag team partner happens. Oh, that's too close. Okay, so these T helper cells, they're going to secrete out what's called interleukins and other cytokines that will help to activate the rest of your immune system. So this is where it helps with activate the B cells. Also helps with bringing in other immune system cells to the area of infection. So attracting neutrophils and natural killer cells using chemotaxis. We'll talk about that later. Attracting our macrophages to come in and eat stuff. And stimulate our T and B cells to go through mitosis. So going through the ability to have a bunch of clone army against that specific pathogens and get them to be fully mature cells to do the job that they are there to do. Okay, so we have our helper cells. Now let's look at our T cell cytotoxic cells. So their job is to bind to the actual pathogen itself. So they're going to target the specific pathogen. So once it targets that specific pathogen, then it does its job, the toxic port. It's going to deliver a bunch of chemicals that are going to do a bunch of different jobs. So we have preferin and granzymes, just like the natural killer cells, to kill off that pathogen. We have interferons, so for specifically for viruses, to keep them from, keep the cells from reproducing those viruses. And we also have tumor necrosis factor, basically to kill off anything that's become a cancerous cell. So once it releases its chemicals, it can go off and do its other job to kill off any other pathogen that it has come in contact with. Then last T cell, we have our memory cells. So they just kind of remain in circulation and they allow for a quicker response because you have a bunch of clones that all are programmed against that specific foreign pathogen. So once upon a re-exposure of the same one, quicker attack, and the person is considered immune because for the most part you won't really have too much when it comes to symptoms because you won't have too much of a actual infection for that second exposure. Then last T cell, we do have some regulatory T cells which help with monitoring and regulating your immune response so you don't do too much and end up possibly killing yourself because your immune system goes in overdrive. So I can't remember which plague infection it was where children and the elderly had a higher survival rate than younger adults. And it was because the younger adults were nice and healthy. Their immune system went overboard and ended up killing off the person too. So those regulatory cells kind of help to keep that in balance and check. Okay, let's look at our other tag team champ. So that is our Bcells. I call them homecoming committee because, yeah, they can do stuff, but they got to have student council or the Tcells help. So these are part of adaptive immunity, but they're the humoral immunity addition. So they're going to develop a specific one adapted to that particular pathogen. But instead of the cells doing the job, it is the antibodies that are flowing in your blood fluid, your body fluids, which is the old word for saying body fluid, are your humors. So for example, like your blood would be one of your humors. So their basic job is to create a army of clones to attack and remember that infection. So you're also going to have a bunch of clone B cells that if you get reinfected, you have a faster response. So their development, born and educated in the bone marrow. So they go to public school, so they don't get to be shipped off to boarding school. But then eventually, once they have matured, then they also get deployed into your lymphatic tissues. Okay, so same thing like your T cells, they have to go through positive selection. So They mature in order to be able to detect a antigen that's been displayed. Then they go through their test. So that's our positive selection. So we're selecting the ones that do good, so that are positive. So if they do not react to a self-antigen, they've passed that test, and then they can be deployed. If they fail, then they undergo negative selection. So they're killed off and no longer able to have that immune response, because if they got out, they would start attacking your regular cells. So with our B cells, so our B cells can work as a antigen presenting cell. So there are receptors on the B cells. They have different structures, so different genetic varieties of B cells. They have different shape receptors. When that receptor binds to a foreign pathogen, then they can become a antigen presenting cell. So they go through that whole process of breaking up the foreign pathogen and displaying just a little piece of that on that major histocompatibility complex. So the AG stands for antigen. So when they display that, the T cell can then read that foreign antigen on their cell surface. That T cell is going to secrete out interleukins. That's going to activate the B cell. So there's our tag team thing situation going on again. So that B cell, once they're activated now because of the T cell, then they can go through clonal selection. So mitosis, make a bunch of them. So a nice clone army against that specific same pathogen. And they'll either become the plasma cells to do the work right now, or memory cells to do the work if you re-expose the second time. So our plasma cells, they're going to create the antibody. So these are proteins against that specific pathogen. So those proteins will bind specifically to that foreign pathogen to activate an immune response. And then memory cells, they just hang out in the body. If you get reinfected second time, or if it's because they were from a vaccine, whatever, you just have a quicker response to that exact same pathogen. Okay, so we're going to start here on another video.